for example, here, there is a gene called CRY1AB. What is this gene? It's a gene, they got this gene from bacteria, Bacillus thuringitis bacteria. When insect, any kind of insect, eat this bacteria, insect die. So they put this protein in the, for example, cotton plant, and you can see if the insect eat the plant, instead of plant, insect die. That's sort of prevention, sort of control of the insect for this cotton plant. I haven't seen before cotton plant, but I plant this one. It is in my lab, and I check the DNA, check the leaf of the plant, and see this gene is in that plant. So it is transgenic plant. Okay, let's finish plant, because usually as a medical doctor we don't like plant. We like human and animals. That's another gene. This gene is deleted, knocked out. It's not transgenic. It is knocked out mouse without myostatin. Do you know myostatin? What's the gene? Where this gene can inhibit growth of muscle. You understand what I mean? GDF8 or myostatin inhibit growth of muscles. So if you are going to be as a bodybuilding, you have to inhibit this one. And this is a mouse which is knock out mice of this one. And this one is, what is this useful for? This is a cow. Can you see that one? Is it good for production of meat?
Yes.
Yeah, exactly. Not this one. This is for production of soja, actually. This is for production of meat. It is not, I would say, you can see, to be honest, it is a real. Just look at this one. If it is open. Look at the size of the car. And it is 1,293 kilograms. Huge.
5 times more than normal cow.
Exactly. It's huge. But you cannot kill this one and eat it as a kebab. You have to grow this one and has some calf and become 100,000 of this one. Because it is very, very expensive now. You have to increase that one.
When you remove this gene, you will get like in the heart, we have a muscle, so it will be more than normal, the muscle of the heart. So it's
all muscles become bigger.
It will not be alive like after a while, after a time.
No, it's alive, it's still alive.
How come the heart is
because the heart is muscle.
Yeah.
Also heart is become bigger.
Bigger, no, you know the like ventricle, when he pump the blood, It's become like larger, and there is no space for blood to go.
It's become larger, and the space become larger, everything become larger. Any muscles, it has any kind of muscle become large. Even the cavities become large.
This is with the growth hormone. When you have the growth hormone,
It's
everything
not
grows.
growth hormone.
Yeah, I know, but this is in the normal size, but the muscle is big. Like when you have some glasses, like in this size, when you put like some.
But the size of cavity also become.
This is with the growth hormone. But when you like just you add more water, and the size is same because you just remove the muscle gene.
Yes, not muscle gene.
Yeah,
The gene can inhibit the
muscle
growth of
growth.
the muscle.
Yeah.
Yes.
So the size of
Maybe, I don't know, maybe it makes some problem in future for this one. But at least 2-3 years more, 3 years. Still, it's the age of this cow. It is alive. The cow is alive. So, I don't know. Maybe in future got a problem. Maybe. But the best thing is look at that and see how can grow up and have a calf and maybe, I don't know.
Maybe.
I don't know about even fertilization of this calf. I don't know. Is it fertilized or not? Can make children calf or not? That's why we have to see. We cannot say now. We have to see. But what I'm saying about genetic engineering. Deletion of a gene make this one do this. Understand?
yes.
At that time, I cannot say about the use of you. In future, you can see the use. Understand?
Yes.
So another usage of genetic engineering is in industry. Do you know this one? What is this?
Sequence.
Clone.
That's
Small clone. Clone is a fermenter. Exactly. What do we have in clone? Bacteria. A lot of bacteria. And we have two bacteria. The good bacteria, which called probiotics. Probiotics. Good bacteria and produce a lot of vitamins. Is it possible to produce this bacteria outside the clone and give it to persons? Yes. You can get yogurt with probiotic, pills of probiotics, and so many things. Yes, it is the results of science and technology and production. You remember? That's the factory, that's the fermenter instead of clone. Exactly. I said 20 years ago, when I came to here, I made this factory and produce, now we produce probiotic in here. And that's good, very good. So I can suggest to you, maybe you can do that. Wherever, in your country or somewhere. So that's another application of genetic engineering, of biotechnology. Why we do this usually, genetic engineering? For better quality and better quantity. Better quantity means production of more bacteria, plant, animal, milk, fish, and so on, and also better quality, to be honest. They do this one for better quality plant as a production of food. Better quality of food. It's one of the reasons we do for genetic engineering. Better quality of the food. Better quality, not only better quality, do you know what it is? It's called. You know that. But it's biofarming. Biofarming is also a name. It's a factory. Production of drug, different drugs in corn. Is it possible? Yes. Corn has got DNA. Can we put one protein of DNA, sequence of the protein, into the DNA and produce RNA protein in corn? Yes, it is. It is possible. So in future, maybe you go to the glossary and say, okay, give me a banana with HPV vaccine. Give me a banana with influenza vaccine, for example. Maybe. I am saying not now. 50 years later, maybe. I'm sure I'm not at that time. I will die, but you are alive, for sure. Biofarming. Is it possible, for example, to put vitamin A in rice? It is. It has vitamin A. It doesn't have. Which one is better? Of course, this one. Is it? What we say to these sort of food, we say GM foods or LMO foods. What's the difference between these two? When we say about the seed of soya, the seed is alive because when you plant the seed, you grow the plant. But the oil of the soya oil, it's not alive. It is GM food. Understand? This one is alive. Bacteria, seeds, they are alive. LMO. And others are GM food. When we are speaking about GM food in the world, we are speaking about trade, government, customer, food safety, and so on. So safety of the food is very important. Labeling of the food is very important. When we manipulate, genetic manipulation of the food, when we do that, testing of that is very important. In the world, we have some administration to check that one, such as FDA in the US, EFSA in Europe, and in Iran also we have FDA in Iran. What I'm going to say about the genetic modified food, when we do genetic modified food, bioethic is very important, so we have to be aware of ethic. But in your life, I would suggest, in your life, don't be not extreme this way, not extreme that way. You should be in the middle, not extremist, but equilibrium, in the middle. It's not from me, it's from Quran. Ummatan wasata means don't be, say all foods from GM food is bad, or not all GM food is good. You have to check it. Understand? Don't say it is bad, absolutely, or it is good, absolutely. No, we have to check it. That's the way between. So, in the middle, not only for food, but also for our life, for our life everywhere, for everything. Okay, enzyme. We have different kind of enzyme that we can produce using genetic engineering. Enzyme for clotting, enzyme for cancer, enzyme for infectious disease, enzyme for genetic disorders. Many enzymes we can produce, and unfortunately already we import them, even in Iran, unfortunately. So we can produce a lot of enzyme. Enzyme is a protein. Enzyme is only a protein. Even you add enzyme to washing powder. Have you seen that? Enzyme to washing powder. Female, they know that. Washing powder. With one enzyme, two enzyme, protease and amylase and others. Industry for enzyme. 2024 is 16 billion in the world. I don't want all of that. I need just 1 billion of that. Is it okay with you? I suppose yes. Enzyme for food. We have a lot of enzyme for food. Biocarbonates, protease, lipase, tenderizer. Do you know tenderizer? For meat. How you can make a meat soft?
With fat, by if there's fat in.
No.
By kiwi.
Oh yes. Why kiwi?
Because
It has protease. If you use kiwi on your hand, you can see your skin change. Or kiwi and some other products such as fish and so on. Kiwi. You know kiwi? You don't know?
بعرف الكيوي لكن سمعنا من الطريقة.
It has protease. Do you know protease? Protease destroys protein in the meat. In the meat. It's tenderizer. Many enzymes we can use for food production everywhere. So, when we are speaking about genetic engineering, it's a challenge. Our life is challenge. Who is the winner? Who is the winner? Okay, let's finish this one and start other session. So what I said about genetic engineering, I said about application of genetic engineering and so on, and finish that. Is there any question about genetic engineering? Because there is a lot of money.
Yes.
Good. You can go if you want. Gene therapy. Gene therapy is another part of genetic. Nowadays is growing up. Nowadays. Before that it was nothing actually. Nowadays it's growing up quickly. I will tell you why. What is gene therapy? Definition of gene therapy. Gene therapy is a medical technology that aims to produce a therapeutic effect through a manipulation of gene. I said, do you remember during the gene therapy, genetic engineering in application of treatment, I said gene therapy. So it's manipulation of gene expression. It is genetic engineering. Or through altering the biological property of living cell. You can change the property of living cell. For which reason? To treat or treatment the patient. In gene therapy, genetic material is delivered to your cell. As I said to you, genetic engineering, you remember? I put the normal gene instead of mutated gene in the genome. in the genome or your cell and change how your cell produced the right protein, the normal protein, not the mutated protein. History of gene therapy you can see from 1990 and 1993, 2000, and so as you can see, comparing to others, gene therapy age for gene therapy, history for gene therapy is about 20 to 30 years. 20 to 30 years. Gene therapy is a technique. Replacement of the gene, replacement of the mutated gene with the normal gene. Just replacement. Is a replacement of that. That's gene therapy. What is the technique? How this technique?
Using a virus or like this.
Sorry?
Using a virus or like this to inject the normal
We use virus, but for other reason, no. Usually, just forget about the virus. Yes, I believe virus is very important, but later on I will explain. Now, we are focusing on DNA because the DNA has got problem here. Look at that. This is a problem, mutation. This is a mutation, cause disease. You want to replace this one. That's the gene. Or you want to knock out the gene. You want to make mutation here. That's the gene therapy. Make mutation or correct this mutation. Correct this mutation. It is mutation. You want to make it correction. This one. Or remove the mutation. Remove the mutation. That's gene therapy. How do you do? How make it this one? In our body, look at this word. Look at this word. Today, I am focusing on usage of nature. In genetic engineering, I use everything from the nature. You remember? I said my factory is the house from the nature. The best is used from the nature. Is there any correction system in our body?
Yes,
Yes, of course. Is it possible during DNA polymerase function? You know DNA polymerase? When the cell divides into two, DNA become two. And DNA polymerase make DNA. Is it possible DNA polymerase make a mistake?
Yes.
Yes. There is a system in our body to repair this one. Proofreading. Oh, exactly. Is it possible we use this property to repair our patient? Of course, yes. Understand? The best thing I advise you, suggest to you, always think, use everything we have around us. The nature. Everything is in the nature. So use that. The nature. When you learn, use it. So what we use exactly homologous recombination. Homologous recombination is for repairment of the mutation. In our body, at the moment, is it possible to use this one by our science and technology as a genetic engineering? Yes. We use our system for correction of our mistake in our body. Is it clear? Is it clear? It's this one. Homologous recombination, which is a technology, it is in our body, in our cells, in our genome already. There is some methods that we use this technique. Long time ago was, for example, Keralox, zinc finger, talent, and CRISPR. I had a student long time ago, a resident. At that time, about 8 years ago, we were going to make a system for gene therapy of thalassemia. We started with zinc finger at that time. And after that, when we working with zinc finger, talent method developed. And when we changed to talent, a few months after that, a year, CRISPR invaded CRISPR system. CRISPR system is very, 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 at the moment, famous method. What are these methods used for? For gene editing, because we wanted to do gene editing. Genome editing or genome engineering or gene editing is a type of genetic engineering in which DNA is inserted, deleted, modified. Look, do you remember the genetic engineering? Cloning, modifying the DNA. Exactly, it is this one. Insertion, deletion, modified, or replace the genome of living organism. Living organism means bacteria, human, our cells, and so on. As I said to you, many methods. Megan nuclease, it's actually forgot this one. Zinc finger, talent, and nowadays they remove these two methods and focus on CRISPR-Cas9. CRISPR-Cas9 is a method of genetic engineering. دکتر من کیش دارم درس میدم دکتر کیشم کلاس تماس بگیرم چی بودش؟ دادم دکتر گفتم مدرسی اونایی که بودن اکرمی مدرسی من یادم گفتم دکتر من حالا بخوایم یادم باشم ستاست دیگه دکتر ولی بعدم بزنم بیام تهران بزنم فردا دکتر بزن مدرسی اکرمی شما بزن دکتر مدرسی اکرمی قدر من اصلا بودن الان بله الان هستن دکتر من و اکرمی هستیم دیگه در خدمتیم بیشتر از دوتا نمی‌پذیرم در خدمتیم دکتر مصطفی So, as you can see here, nowadays we are focusing on CRISPR-Cas9. This method is, to be honest, revolutionary technique in genetic and in medicine, for sure, revolutionary. It's changed everything in medicine. It is changing. Check database. Ask AI. You know AI? Good, very good. Don't ask me. Ask ChatGPT. Ask CRISPR and Cas9. This method, do you know where is this method coming from? Do you know? It's funny. it is funny. I'm saying funny. This method is coming from yogurt. 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 Yogurt.
Yogurt.
Changing all medicine. A method from yogurt. Lactobacillus, a bacteria. Do you know yogurt? Yogurt. Must. It's coming from lactobacillus. This system, this technique is sort of immunity for bacteria. Immunity system for bacteria. Understand? And somebody found this one. What is this method? This method actually make when virus go to the bacteria, phage. I don't know whether you know phage or not. It's a bacteria virus. When go to bacteria, this system, CRISPR, a protein. It's a protein. This protein cut the phage virus. Cut the virus. Why? Because destroy the virus. It's sort of immunity, defense system for bacteria. And people use this system, defense system, in human. Understand? For what? For repair the DNA. For repair the DNA. Because the problem for DNA, how many base pair we have in the genome? How many? 3.3. And one mutation, imagine, to find one mutation in 3.3, 10 to 9 nucleotide. Look at that here. How many 6 are here? How many? About 100? More or less. 100. Imagine, instead of 100, we have 3.3 to 10, 9 seeds. And we are going to find one mistake, one person in these people. Is it easy?
No.
No, not at all. It's very difficult. You are going to find one mutation in 3.3, 10 to 9. One mutation, C change to G in this DNA. Imagine, it is very difficult. From the beginning of DNA, you have to go, see? No, you can't. You can't go and check it this one. And CRISPR system can do that. This protein move on DNA and find exactly that mistake and cut it. Understand? Understand? Is it clear? This protein sit on DNA, move on DNA, and find exactly the mistake and cut the mistake. And our system, our repair system, make that one, the mistake repair. Correct the mistake. Is it clear to you? So that's the system. I mean, the nucleus here is CRISPR, Cas9, the protein of Cas9. It is the protein. This protein stick to DNA, and go along DNA for 3.3 multiply 10 to 9 nucleotide. And find, the mistake here. Cut it, the mistake. That's and use our human repair system and repair that. Is it easy? No. No, it's very difficult to find that. And it is, this protein is not belong to human. It was belong to bacteria. And the person, look at this one. That's the system of CRISPR. And the person who found this one got Nobel Prize 2020, 4 years ago. Got Nobel Prize, these two persons. Do you know why? To find this one. Let's take the DNA and go. To find the Cas9. That's Jennifer Doudna and Charpentier. And two other persons also, they did a lot about CRISPR, but they didn't got Nobel Prize. I don't know why. But these two persons, they got Nobel Prize just for CRISPR. And CRISPR changed gene therapy revolutionary. Day by day, you can see treatment of disorders starting to develop. Before that, it was not. 6 years ago, I published a paper in Iran for CRISPR, and my students working on CRISPR in Iran. It was the first paper about CRISPR in Iran. It's about gene therapy and some other, I said thalassemia and so on. CRISPR applications. So, what I am saying now is about gene therapy. The method for gene therapy is gene editing, and the method and the protein which we use for gene editing is CRISPR. Is it clear? Is it okay? So, what is the application for CRISPR? CRISPR is a method, and the protein is Cas9. Look, the protein is Cas9. CRISPR is a method of checking the DNA to cut it. Which application? Therapeutic application for CRISPR-Cas9. Therapeutic application for cancer, liver, viral infections, Huntington or Duchenne muscular dystrophy, blindness, and mitochondrial disorder, genetic bloodline disease, and so on. Many disorders is adding to this one using CRISPR-Cas9. Is it revolutionary? Yes. It is increasing, using application of gene editing using CRISPR system and Cas protein is increasing every day. Several important issues about CRISPR-Cas9 we have to be aware of that is ethical consideration, safety, and off-target. Do you know off-target? No. Cas protein stick to DNA and go through the DNA. 3 multiplied 10 to 9 nucleotide. Just by mistake, if find exactly the mutation and cut it is okay. If by mistake cut here, is off target. Understand what's the meaning of off target? By mistake cut here, not cut the mutation. Understand? Okay. A regulatory challenge is another one. Another thing which is very important you have to know about that is immune reaction. Cas9 is not belong to human. It's not a protein of human. Do you know when you inject a protein, foreign protein to human, what's happened?
Immune response.
Immune response. We produce antibody against that. Cas9 is protein belong to human or bacteria? Bacteria. So when you inject Cas9 to human, it makes antibody against that one. So we have to be careful about that one. Because when you stick to the DNA and go through the DNA and find the mutation and cut it, you have to destroy the protein. Otherwise, it makes antibody against the protein in our body. Technical limitation, social implementation or implication, future direction, and many things you have to be aware of this system, CRISPR system and Cas9. You must read this one. Okay, back to gene therapy from the method of CRISPR. Back to gene therapy. There is another system for gene therapy which is very important, is immune therapy. Immune therapy is another method of gene therapy. There is a gene. Do you remember TSG18 I said to you? What was that gene from? I showed you TSG18, a gene which I found from testes. Do you know there is a special immunity in testes?
Yes.
Or in brain, or in eye? Do you know when something happened to our eye, go to this eye, they have to remove the eye. Do you know why?
Because if the barrier is disturbed, the immune system attacks the eye.
Because we have a barrier. Exactly. We have barrier in our eye, and our protein in our eye, human eye, they are not known for our human. And if exposed the eye, the protein in eye, exposed to our human body, our human body, our body make antibody against our eyes, and destroy another eye. That's why they have to remove the eye quickly. Otherwise, we have blindness in another eye. Where do we have barrier? Brain, blood barrier. Do you know blood barrier? Brain blood. And also we have testis, blood barrier. In testis also we have barrier. Stem cell, do you know Sertoli cells?
Yes.
Sertoli cells. Stem cells and sperms producing Sertoli cells in seminiferous tubules between Sertoli cells. Germ cells and Sertoli cells are in seminiferous tubule. Seminiferous tubule, there is no blood in the seminiferous tubule. If we break down seminiferous tubule, what happens? Such as this one. With testis. Very, very hard bit to testis. Some of these seminiferous tubules break down. And our germ cell and sperms exposed to blood. What happened? Our body produce antibody against our sperm. Against our sperm in human, in male. What happened? What happened? Infertility. Infertility. Make infertility. Understand? Because the sperm stick together cannot go through to oocyte, make infertility. Even during the period, if human has got mating, a sperm exposed to female blood, female produce antibody against sperm and makes infertility. Understand? That's immune system. If we have this sort of gene in cancer, testis specific genes in cancer, not only in testis, in cancer, our body produces antibody against that one. So it is immune therapy. Understand? Immune therapy. Immune therapy is this gene, TS310, MAG, and many other genes used for immune therapy because they are in cancer also, not in testis, but also in cancer. Specifically, not in our body, not anywhere in our body. Okay, let's back again to clinical trial in gene therapy. You can see clinical trial in gene therapy in 4 different: cardiovascular, neuroscience, ocular, inflammatory, and so on and so on. This part is immune therapy for gene therapy. This one is tumor suppressor gene for cancer, and so on. Let's go for another. Do you know P53? Do you know P53 or not? What is P53? It's tumor suppressor. Is it right?
Yeah, I wanted to say if
Okay.
there's a mutation and then we have to...
Exactly. It is tumor suppressor. But which kind of protein is this tumor suppressor? Look at this one. Which kind of protein? Which kind of protein? Look, this is a growth inhibitory gene. Growth inhibitory gene is this one. So, it is growth inhibitory gene, and P53 is stick to here, upstream of the gene. What is upstream of the gene? Is promoter. If one protein stick to promoter, is the name of that is, what's the name of that? What is the name of that? Transcription factor. You know transcription factor?
Yes.
It is transcription factor. P53 is transcription factor. Understand? I gave you 2 exam questions. This is another exam question. Another exam question was between fish and carnivore. Do you remember this? I give you the answer. I give you the question also. This is on you. So, P53 is transcription factor. Stick to here and produce inhibitory growth factor. Understand? So it is. Is it possible to put this one, use this P53 for destroy tumor?
Yes.
This is, we call to this procedure gene therapy. Gene therapy of cancer. Different kind of gene therapy do we have? Somatic cell transgenesis, gene therapy for somatic cell, gene therapy for cell line, germ line. It is inhibited, actually. It's not easy. We have 2 sort of gene therapy. One is ex vivo gene therapy, means we do everything out of our body and then inject to our body. In vivo is anything happen is in our body, in the cell. What's the first gene therapy? What was the first gene therapy about more than 20 years ago? On which gene? What was the first gene therapy? Skill. Severe combined deficiency. This person, 20 years ago, born without ADA, adenosine deaminase gene, has got mutation here. So the problem with this one was severe combined deficiency. It means this girl cannot stay out of the bubble. Do you know why? Out of the bubble we have a lot of germs, virus and so on, and die. So in the bubble is everything is straight, completely, without germs. So after that, look at this one, that's the living, should be in the bubble. After gene therapy, out of the bubble. How did gene therapy actually ADA gene? Do you remember ADA gene?
Yes.
This gene? It has mutation, actually replaced with normal gene, not mutated. Replaced with normal gene. Gene therapy is not easy. They have severe immune response. Sometimes when you're using adenovirus, when you're using retrovirus, it is we have leukemia-like. These two persons died due to gene therapy long time ago. So I will explain why these two died. Okay. So for gene therapy, two problems happen and very serious. One is immune response. Second is random insertion. It means somewhere we put the gene, we have to replace the gene, this gene, replace this gene, but sometimes this not replace here, replace somewhere here, the wrong position, and it's make problem. That's a random insertion. Okay. Somebody asked me here about told me about virus. Now I want to say, what is the most important issue in gene therapy? Is gene delivery. Can you explain this one?
Taking the gene, inserting it into the DNA or RNA where we want, to edit. We want to do gene therapy on an animal, so it means gene delivery means inserting the gene into that animal.
When you are speaking about DNA, where is the DNA in the cell?
The nucleus.
The nucleus. We discuss a lot about mutation in the genome. We discuss a lot about mutation in the genome. So, when we discuss about mutation in the genome, where is the genome? Genome is in the nucleus. Nucleus is in the cell. Look, if somebody wants to kill you, first the person should come to the university and then come to the class and then kill you. So there is two barriers. Am I right?
Yes.
Kill you after two barriers. If somebody wants to repair a gene, somebody come and say something to you. first come to university, enter to the class, and go to you and say blah blah blah. Am I right?
Yes.
So, what's the most important issue for gene therapy? Coming into the university, coming to the class. Because they can come to the university, come to the class, and then find you in this class. Because many people are here. Understand? Imagine you are a mutation. You are a mutation. The first, for gene therapy, the right sequence, I mean the gene, the right gene, should come to the university and come to here and replace you. Am I right?
Yes.
So, what's the main issue is entering or delivery. Gene delivery is the most important issue for gene therapy. How we do that?
My viruses.
We can use viruses as a
Delivery.
vehicle. As a vehicle for delivery. Am I right? Because virus knows the door. But virus doesn't know about the seed. CRISPR knows about the seed. Am I right?
Yes.
CRISPR knows. CRISPR can find him and replace him. But the first is how CRISPR comes here with the gene. CRISPR has the gene and comes inside. But how? You have to put the gene inside a vehicle. What's the vehicle? Virus. Understand? Why we say virus? Why? Why virus? We have different type of gene delivery. You know which type of gene delivery? Physical, microinjection. Is it possible to inject by needle into the cell? It is possible. But is it possible to use microinjection for gene therapy?
No.
How many cells do you have? Billion cells you have in your body. Okay. Million days or years you have just to inject cells, because billion cells you have to inject DNA. It is impossible. But virus can do that. Virus can go to all cells and enter to cells. Am I right? Okay. So we have another vehicle. I am saying vehicle. That's electroporator, microinjection, gene gun, liposuction, polymers, liposuction, and so on, and the virus. Many gene delivery methods. Some of them are this one, and this is the viral. Which one is the best for gene delivery in gene therapy? Is this these two. Look at these two. What are they? Is adenovirus and retrovirus. Understand? This is adenovirus. And this is another method, such as liposuction. Do you know liposuction? Do you know the cell wall? The cell wall is bilayer of lipids. You know that? Bilayer of lipids. We can put DNA in a vesicle of bilayer of lipids, and this one fuse to the cell wall and put the DNA inside. That's gene delivery. But not as functional as virus. This is when I was in Canada, I used to produce virus. This is, can you see these are cells? Look at these. Many cells are here. One cell, two cell, this is cell. Many cells are here. Look at this. For this one, I use liposuction by layer lipid to transfer DNA. Do you know GFP? You remember GFP? Green fluorescent protein from fish, starfish. So I put this gene in the liposuction lipid and transfer to the DNA. Look at that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Just about 20 cells. I delivered GFP to 20 cells because under UV it is green. You remember green? Okay, let's see. I put this gene in adenovirus. This one. This is adenovirus. I put the gene in the adenovirus and with adenovirus send this one to cells. How many cells are they have GFP? Can you compare these two? Which one is more efficient?
Bottom.
This one or this one?
This is the bottom one.
Of course this one. It is much more efficient. Now you can understand why we use virus as a vehicle to deliver DNA to cells. Understand? Okay.
Viruses have a specific, like when you have a bone marrow cancer, there is a special virus to attack this bone marrow.
Why? Because of
Specific.
I tell you why. Just 2 slides later. I tell you. Which viruses we use already? We use retrovirus, adenovirus, adeno-associated virus, lentivirus, and some other virus. Virus for gene therapy act as a what's the, do you know adenovirus? You pass microbiology?
Yes.
What is adenovirus?
HIV.
Adenovirus, not HIV. I am saying about adenovirus. What's adenovirus? Cause what? You know which disorder, which infectious adenovirus produce?
We forget. We forgot that.
Adenovirus produce what? Which infectious?
Which cavity?
Why? Adenovirus produce
We know about it.
What?
It causes HIV.
HIV is HIV virus. No, adenovirus produce gastrointestinal diarrhea or bronchitis. Bronchitis. Understand? Adenovirus is an infectious virus. Can infect your body. So, what is the difference between adenovirus, which I use it for gene therapy? I produce this adenovirus. What's the difference between this adenovirus and the adenovirus you get from your friend?
This is not
This
You know,
doesn't cause disease.
The most
They can
infectious virus which we bring from Hajj, Saudi Arabia, during the Hajj, we bring the virus in our countries is adenovirus. It's adenovirus. Diarrhea, you know, it causes diarrhea. It causes bronchitis. It does also infectious in eye, any epithelial cells as we have, such as conjunctivitis. Do you know conjunctivitis?
Yes.
Yeah.
Your eye. So, in this situation, that's my question. What's the difference between the virus here, I use it for gene therapy, and the virus, natural virus, which can cause bronchitis, cause diarrhea?
You remove the virulence factor like basically. You remove the virulence factor of the virus.
We remove virulence. What is the virulence factor? You said right.
Like a protein,
No, no, I
some,
don't tell
some genes.
Yes, some genes, but which genes?
It's like that have the virulence factor.
But
the virus which is infectious, they use the genetic machinery of host and make these proteins to cause infection.
yes, they have transcription factors.
Yeah.
You want to say virus has got transcription virus, while transcription factor can hijack the system of the cell.
Yes.
Okay. Hijack the system of the cell and produce what?
It's DNA.
Produce virus. Produce virus. And produce virus and break the cell.
Yes.
What's the virulence of the virus? The virulence of the virus means production of the virus in the cell. Understand? And destroy the cell. So, if I remove a transcription factor from the virus, cannot replicate. Cannot replicate. It would be a vehicle. Understand? In this situation, the virus is just vehicle. Enter to the class. That's the vehicle. Go to the cell, but not replication. Understand?
Yes.
There is no replication for the virus in the cell. It is engineered virus. Understand? What is your classroom? Is about genetic engineering. You remember I said about genetic engineering produce virus? Yes, I used to produce this virus and I used to sell. Each virus I used to sell about $2,000 in Canada. I used to produce virus, purify the virus and sell it. And got $2,000 for each vial. That's genetic engineering. Understand? That's why I said genetic engineering is money. So, we have different, you must memorize this slide. Which kind of virus we have for gene therapy? Usually retrovirus, lentivirus. Which one is a small, which one is large, which one makes inflammatory, such as this one, for example, adenovirus make inflammatory for body. But retrovirus, as you can see, they don't make inflammatory in our body. But retrovirus integrated into the genome, but adenovirus cannot integrate into the genome. This is adenovirus here. Can you see the adenovirus? That's the schematic picture and it's the natural picture. They have knobs. What is this knobs? This knobs. This you have to learn this sentence. All viruses stick to the cell through a receptor. Through the receptor. You said exactly right word. We have some virus for some tissues. Because the receptor of the virus is in a specific tissue, on a specific tissue. Is it clear? So, adenovirus stick to epithelial tissues. Lentivirus stick to, what's the origin of lentivirus? What's the father of lentivirus? You know. You know. What's the father of lentivirus? You said many times.
HIV.
HIV. Yes. HIV is father of lentivirus. They changed the HIV and make lentivirus. So, where is the receptor of lentivirus? Bone marrow. Because HIV target bone marrow. So it is for bone marrow. As you can see here, we deleted E1 and E3 in the virus, and the virus doesn't replicate in our body. It works as just vehicle and stick to receptor, transfer the gene into the cell. Look at this. Stick to the receptor and transfer the DNA as a vehicle into the cell. Understand? Yes. That's intra. Gene delivery of the virus. Look at this one. So the question is this one. We deleted the gene for replication of the virus. How we can produce virus? You didn't ask me.
What is the question?
Because the virus injects its genetic material into the cell, and then the cell, when it reproduces the virus genetic material. Right?
I said I deleted E1 and E3, which are transcription factor, gene from genome of the virus. So this virus cannot replicate in the cell. It is just vehicle. So transfer the DNA gene into the cell. How can I produce this virus? I have to replicate to sell that one. I said I used to sell the virus.
Replication of the cell.
PCR.
Change your word. Not mutation.
I mean mitosis of the
No,
cell. We
no.
induce the virus.
No. I deleted E1, E3 from the virus. Is it possible?
We have E1 and E3 in our cell.
We don't have. We don't have.
Put them again.
Not to the virus. You make these two together. Not to the virus, to the cell. Is it possible I put E1, E3 in one cell line?
Yes.
It is. I put E1 and E3 from the virus, take out, and put in a cell line, genome of a cell line. This cell line would be the factory for viral production. You remember I said factory, cell factory. Understand? This cell line is a factory for virus production. It is this one. This is my factory. I used to produce the virus. Do you know what this is? This is full of the virus. Thousand virus in this cell. I extract the cells, these cells, and break the cell wall and purify the virus and sell it. And got $2,000. Is it okay with you? Do you like that?
Yes.
Of course. This cell line, which is HEC293, has got E1, E3. Not mutation, E1, E3. You said right, but not mutation against E1 into E. Is it okay? In the cell line, as you said. So, this cell line would be the factory for production of the virus. Is it clear? And this is the cell ready to produce, ready to kill, ready to break down and get the viruses. Understand? This is the production of the virus. What's the future of the gene therapy? That's then. Sorry?
Target specific.
Target a specific gene therapy. Can you give me example and finish it? Give me example.
It's like when you change the color, there is a gene
No.
that will replace it.
Easier, simple. Very. Do you know breast cancer
Yes.
is one of the most common cancer in female. Breast cancer doesn't kill anybody. Oh, no, kill. We know kill. No, I say breast cancer itself doesn't. What is the cause of this from
Metastasis.
metastasis? Exactly. Metastasis. Not breast cancer, because you can remove the breast. Okay, finish. Not finished. Because one cell go to somewhere else we don't know where.
Then
We don't know where. How you can find this cell in our body? That's it. How? How? Give me, please activate your gray material in your brain. Please activate that one. If you want money, if you want something, activate gray matter. Please. You don't want to use it? Gray matter.
Just your question.
The question is how you can find a metastatic cell in our body. Do you remember Jules Verne movies? Some very small machines go to our body in the blood and go find it. It's like that. How? Is it possible use virus? What I said adenovirus stick to. I said to you, adenovirus has got receptor. It is receptor. This is receptor of adenovirus. This one stick to
Cell.
what?
Cell.
Epithelial cell. Is it possible for me to change this one? A protein. Change this virus. Stick not to epithelial cell.
Metastatic cell.
Stick to cancer cell.
Yes.
Is it possible? Yes, it is. Understand? I can find something which is only in cancer cell, on cancer cell, not anything else. So I put antibody here, and this one goes to the cells, to our body, and stick to the cancer cell. Is it possible?
Yes.
Is it possible for me to put a toxic, do you know toxic? Toxic gene inside the virus?
Yes.
What would be the result? This one goes and find the cell, stick to the cell, put the toxic gene into the cell. What would be the results? Cell die. Is it? If I put toxic gene in the virus without changing this one is bioterrorism. Because I kill everybody. I kill myself and others. Am I right? If I put toxic gene here and change this one is target gene therapy. Understand?
Yes.
Understand? That's why I'm saying use gray matter. Use gray matter. It's target gene therapy for type of cancer treatment and many things. It means you destroy exactly target specific cells. Understand? It is clear. It's also it is. Look at this one. Put antibody against the cell. Exactly what I said. It's from Nature journal. It's from Nature journal. It's not from me. It's not from my gray matter. It's from other people. Okay. Any question?